Hi. So in the previous presentation, we learned about orthomyxovirus. We learned about influenza over there, something which comes under the superfamily Myxoviridae. So Myxoviridae are a class of viral species or viral family which has some predilection towards the mucin receptors present in the on various cells, and they also show some kind of attraction towards the mucosal lining. So no matter whether it is an orthomyxovirus or paramyxovirus, all the members show some sort of relationship or some sort of attraction towards respiratory tract, and most of them can appear or can come to our body through respiratory route or through inhalation. So in this presentation, we'll be focusing mainly on the different members or the various types of members coming under the superfamily Paramyxovirus. So let's jump into the presentation. All right, so it comes under the superfamily, superfamily Myxavirus. Myxavirus are those virus which has got affinity of virus towards mucins, and it is composed of two families, Orthomyxoviridae and Paramyxoviridae. We have already compared their basic characteristic features in the previous presentation where we learned about Orthomyxovirus. So the basic difference you should always remember while comparing orthomyxovirus from paramyxovirus is that orthomyxovirus has got segments of RNA. Both are RNA containing virus. They are RNA virus. Okay, so the speciality of the genetic material for orthomyxovirus is that they got segments of RNA, and with the help of these segments of RNA, they can rearrange their genetic material or genetic makeup, so they can come up as a new strain every time whenever they appear as causing pandemic, epidemics, or even any localized infection. So they can change their behavior, they can change their genetic makeup and come up as a new variety or a new strain. That's really dangerous for us, because that will create serious issues for effective diagnosis and effective treatment as well. So we don't have any super vaccines or common vaccines available for orthomyxovirus. But when you talk about paramyxovirus can be a little larger than orthomyxovirus. They show some kind of pleomorphism in terms of their shape, but they consist of a single linear RNA strand as genetic material. So that's quite stable. So it won't change upon generation, it won't change upon generation, just like the way how orthomyxovirus will do. So somehow we managed to tackle paramyxovirus by some effective vaccination or successful antiviral drugs. So paramyxovirus are less dangerous than orthomyxovirus, but we should always consider the status of the individual who is getting paramyxoviral infection. Most of the paramyxoviral infections primarily affect young children or people who are in the old age conditions, people who are immunologically weak. In those situations, it becomes really serious, right? It can develop into a serious complication. So we should consider all those things in our mind. So okay, this is just a basic comparison. So let's jump into the actual paramyxovirus. Yeah, this is one more slide which actually compares the structural features of this is orthomyxovirus, and here we got a paramyxovirus. Orthomyxovirus is quite sophisticated, and they got a highly developed envelope. They got a matrix protein which is also present in case of paramyxovirus also. Here we have this envelope, or here we have the envelope. Here they got this matrix protein, and surrounding this matrix proteins or internal to this matrix protein, they got the capsid, which is composed of a capsomere, but which is lined by a long strand of viral RNA with large numbers of ribonucleoproteins. Three types of proteins are there: nucleocapsid proteins, large proteins, and phosphoproteins. So three types of proteins are attached with this RNA. So that's a single stranded RNA. Whereas in case of orthomyxovirus, they consist of almost 8 pieces of strands of RNA, so which is helping them to make successful variations or changes whenever they make a replication, so they can easily change the genetic makeup and come up as a new strain. Now let's talk more about the structure of this paramyxovirus. They have a nucleocapsid which is surrounded by a lipid envelope which has matrix M proteins and its base, and at its base, and two types of transmembrane glycoprotein spikes at the surface. So they got nucleocapsid, and this nucleocapsid is covered by another protective layer known as matrix proteins, and surrounding this matrix proteins or matrix layer, they got this envelope. Envelope is actually part of the host cell membrane. And overall the surface spikes of this paramyxovirus starts from the matrix protein layer of this virus. That's the point. So matrix protein layer harbors, they act as the base for the two types of transmembrane glycoprotein spikes. So this is one type of spikes and this is another type of spikes. So basically in orthomyxovirus they also got two types of surface spikes. One is the N spikes or neuraminidase activity, and another one is H spikes, hemagglutination activity, but both are originating from the envelope. They won't reach till the matrix region. But in paramyxovirus, these two spikes, instead of N and H spikes, they got fusion spikes and H spikes or G spikes for the attachment. So these two are the important surface spikes present in this paramyxovirus, and both these spikes are originating from the matrix proteins in case of para-influenza virus. So the longer spike is called hemagglutinin spikes. This is the hemagglutinin spikes or G spikes, which may also possess neuraminidase activity. So this single hemagglutinin spikes has got two activities. It has got hemagglutination activity as well as neuraminidase activity, whereas in orthomyxovirus these two are supplied by two different surface spikes, whereas in paramyxovirus it is done by, the functions are done by or handled by same surface spikes, neuraminidase and hemagglutination activity. So I can call it as H spikes or HN spikes or G spikes, which is required for the initial attachment with the host body. So apart from having this G protein spikes, which is quite larger than the second spike, this is F spikes or fusion protein. And this F protein or fusion protein is responsible for the fusion of the viral envelope with the plasma membrane of the host cell, and it is essential for infection. So if you remember the various steps in viral replication, virus has to attach to the host cell membrane. The initial attachment is happening with the help of this G spikes or HN spikes in case of paramyxovirus. That's adsorption process. After the adsorption process, next is the penetration process, where this virus need to introduce or insert their nucleocapsid into the host cell cytoplasm. So for the effective penetration, the viral material, viral outer covering should fuse with the host plasma membrane, and that fusion is happening with the help of this F spikes. I'm talking exclusively about paramyxovirus. So this is the function of F spikes in paramyxovirus. Initial interaction happens between G spikes and the host body, followed by the fusion of plasma membrane is happening with the help of fusion proteins or F proteins present in paramyxovirus. And F proteins are also helping in cell-to-cell fusion, causing large giant cell formation or syncytia formation. So if you see the cytopathic effects of paramyxovirus, these paramyxovirus can cause the fusion between adjacent cells, and the cell become a large multinuclear cell with a single common cytoplasm having plenty of nuclear material, and that fusion is happening through F protein. So F protein has got two functions. Function number one, it will enable this virus to interact with the plasma membrane of the host body to initiate penetration process. And cytopathic effect, it has got an impressive cytopathic effect which is responsible for the syncytia formation. Syncytia formation means it's a giant cell formation which is formed by the combination of different cells, which is having a common cytoplasm and plenty of nucleus, multinucleated giant cells. Now, talking more about the paramyxo superfamily, paramyxoviridae. Viridae always stands group of viridae means a group of virus. So this paramyxoviridae superfamily has got two subfamilies. One is paramyxovirinae and pneumovirinae. So paramyxovirinae and pneumovirinae are subfamilies of paramyxoviridae. Let's see what are the members which is coming under the paramyxovirinae. So the important members coming under paramyxovirinae includes respirovirus, parainfluenza 1 and 3, rubella virus, which causes mumps and parainfluenza 2 and 4, mobilivirus, which causes measles, and Henipa virus, which causes Hendra and Nipah virus. All those comes under paramyxovirinae subfamily of Paramyxoviridae. So, the second subfamily, Pneumovirinae. Pneumovirinae includes two subgenus, Pneumovirus and Metapneumovirus. So, Pneumovirus consists of respiratory syncytial virus, which is actually causing the giant cell formation, giant syncytial cell formation. And Metapneumovirus that genus includes the human Metapneumovirus. We'll see about each one of them in the following presentation. So let's begin our learning process with knowing about mumps virus. So mumps virus comes under the rubella virus family. Yes, rubella virus family, rubella virus genus of the subfamily Paramyxovirinae can cause mumps. Right, and this is the typical clinical presentation of mumps. So mumps is characterized by the non-suppurative enlargement of the parotid gland. So in this image you can clearly make out that this child has got severe swelling at the parotid gland, which is a non-suppurative complication, and it is caused by rubella virus. And the morphology of the virus is exactly the same, which has got HN spikes or H spikes and F spikes. They got a single-stranded RNA with 3 combinations of ribonuclear proteins. They got a nuclear capsid, and they got metric spot. In structural wise they are almost same. So mumps virus is a typical paramyxovirus which possesses both HN and F protein. So yeah, this is the classic structure of paramyxovirus which causes mumps. The virus agglutinates the, it can agglutinate with the erythrocytes of various origins, and hemagglutination is followed by hemolysis and elution. So this can combine with the RBCs or hemoglobins, or even RBCs of various origin, and so it can lead to severe clumping of RBCs, can block the areas, or block the arterial blood supplies. That's the specialty of this mumps virus. Now, talking about the cytopathic effects associated with the mumps virus, it consists of a syncytium formation at the presence of acidophilic cytoplasmic inclusion. It will produce acidophilic cytoplasmic inclusion with the syncytial formation, especially at the parotid gland. And the source of infection is usually aerosols. Just like the way I said, most of the paramyxovirus or myxovirus, the route of entry is usually through aerosols. And this aerosols which is coming from an infected person, the person who coughs or sneezes, he can spray or he can spread plenty of aerosol, which is composed of plenty of viral particles, and it can reach another healthy individual through inhalation, and probably even through conjunctiva it can reach there. And very occasionally there will be direct contact through direct touching, or even through saliva or urine. These are the common agents which can transmit the infection from an infected individual to another healthy individual. And the incubation period varies between 12 to 25 days. So what actually happens to the virus is, virus will reach the upper respiratory tract, of course through inhalation. It will reach the upper respiratory tract, and it will start its multiplication at the cervical lymph node. So they have to multiply to increase their number, and from the upper respiratory tract lymph node they will disseminate through bloodstream, causing primary viremia, and from there virus can induce to various organs through blood circulation, including our parotid gland, where they will multiply, damaging them, syncytial formation with severe non-suppurative swelling over there. And the common clinical feature will be the common clinical feature for mumps infection will be the parotid swelling. That's the first sign of swelling, and there will be prodromal malaise. There will be some kind of malaise condition, and usually parotid swelling is unilateral in the earliest case, but it becomes bilateral within days, because the parotid glands of both the sides will be affected. It is always accompanied by fever, and there will be lock-up, severe pain, and the baby will not be able to lift his neck, and he will not be able to chew properly or eat or drink properly. And this is usually an infection which is commonly seen among young children whose immune system is very weak or developing, and there is a high chance among these age group to get this infection at a very faster rate. So it occurs as an epidemic in children between the age of 5 to 15 years of age. It is transmitted by droplets, direct contact, fomites contaminated by saliva, even urine. We discussed about these things. Now, talking about the clinical features, there will be unilateral swelling at the earliest stage, but it can develop into bilateral swelling associated with a fever, pain, tenderness, oversight over the site affected, and it is usually a non-suppurative complications. Apart from this, there will be other associated complications like epidermolysis, where it is commonly seen in 1/3 of the population, especially in male patients. And in epidermolysis, the testis becomes really swollen up, and it is usually accompanied by fever and chills. They will be painful, and if you make a delay in proper treatment, that can lead to the sterility in those cases. It can lead to orchitis, usually swelling at the testicles, even meningitis, meningoencephalitis, and less common complications includes this arthritis, ophoritis, nephritis, pancreatitis, thyroiditis, and myocarditis. All these things are happening because the blood is carrying this infective agent to various areas and various locations. So wherever it can go, they will stay there. They will start its multiplication, causing localized to severe multiplications, localized to severe swelling over there. Now, talking about the lab diagnosis, we can collect a different range of samples or specimens. Starting from saliva within 4 to 5 days, urine up to 2 weeks, we can take the urine sample. Of course, we can take CSF sample up to the 9th day of infection after the onset of illness, and these are the common samples, and we can isolate this virus in different culture plates. We can we can isolate we can grow them in lab conditions. We can inoculate them into different cell lines like monkey kidney cell cultures or human amnion or HeLa cells, and growth detection is usually done by heme adsorption inhibition by specific antisera. We can also go for immunofluorescence, which is a rapid test, which is a rapid method, but it requires the presence of fluorescent microscopy. Of course, we can go for other immunological serological examination, where we'll directly target the antigen by immunofluorescent assays or even by ELISAs. And in serological examination, there will be an increase in titer of antibodies. In paired serum, there will be definitely increase in antibodies in serum, and of course the antibody presence can be identified in cerebrospinal fluid, and especially IgM antibodies. IgM antibody level will increase because IgM will be the primary antibody which is produced earlier at the time of infection. Of course, we can go for polymerase chain reaction. That's a most modern molecular techniques to diagnose this clinical presentation. Talking about the prophylaxis and treatment for this vaccination, for this infection, we can cure this condition by taking live attenuated vaccine known as gerillin strain. Right? They developed this kind of strain of the mumps microbe. What they did is, like, they attenuated this this viral particles in the eggs, embryonic eggs of chick and chick embryo fibroblast culture, and they repeatedly subculture it and until it become attenuated. So that can be given as a vaccine. That's a live attenuated vaccine, and recommended usage is only after 1 year of age. It should not be given below 1 year of age, because maternal antibodies may interfere with the multiplication of this vaccine virus. So it should be given above the age of 1. So there will be some contraindications for giving this vaccine at the time of pregnancy, immunodeficiency and hypersensitivity condition. We should avoid this kind of vaccination, but it can be given along with measles and rubella vaccine, like triple vaccines, MMR vaccine, measles, mumps, rubella vaccine. It provides effective protection at least for 10 years. MMR vaccine is susceptible, and it's working for it will work for another 10 years.